Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGiorgis. Today is part 2 of our discussion of the paper titled Kinesin 3 is an organelle motor in the squid giant axon. It was published in Traffic in 2008 by myself, Tatiana Petikova, Teresa Evans, and Thomas Rees. Last time I mentioned some of the sequence work that led up to this particular publication. We talked about the central dogma in which DNA is transcribed into RNA and RNA translated into amino acid sequences or proteins. And it turns out in Ron's original study, the only thing he really had available to him was an opportunity to biochemically purify the protein, which is not easy. And he did a really wonderful job of purifying it and then characterizing conventional kinesin. Of course, you know about the genome. The genome is sequencing all of the DNA of an organism, that is, all of its chromosomes. And I wanted to sequence just the RNA molecules because the RNA molecules tell you what proteins are being expressed in that particular tissue type. And it's easier to isolate RNAs and sequence them in large numbers than it is to do the genome or to try to get after proteins through biochemical purification. Through this process, I was able to sequence 22,000 different RNA molecules, and I was able to identify what each of those RNAs encoded, that is, what proteins it would ultimately generate. And it turns out there was a set of RNAs that encoded 6 different types of kinesins. One of the kinesins happened to be the one that was isolated by Ron Vale and his colleagues, conventional kinesin, and the other 5 were newly discovered kinesins that are expressed in the cell bodies of the squid giant axon, along with Ron Vale's kinesin. To try to study the function of each of these motor proteins, I generated specific antibodies against each of them. And in this paper, I focused on one of the 6, which is kinesin type 3, kinesin 3 in squid. And I compared kinesin 3 to conventional kinesin or kinesin 1 because I was still interested in what was happening in the squid giant axon. There were a number of issues. We said that purified kinesin that is kinesin 1 transports latex beads at 0.5 micrometers per second, while organelles, or vesicles, if you want to refer to them as that, isolated from squid axoplasm, travels at about 1.6 micrometers per second. So one problem was this rate issue. The second was that the Western blot that Bruce did, to me, suggested that only 0.1% of the kinesin is found in the organelle fraction, while 99.9% is in the cytosol, or the S1 fraction. Same thing, same different name for the same thing. So that didn't sound to me like kinesin 1 was responsible for the transport of the organelles that move along the microtubules towards their plus ends. And finally, my experiment of trying to label organelles with kinesin 1 antibodies didn't work. So we had three different problems: the rate, the fact that kinesin is found in the supernatant or the cytosol or the S1 fraction and not in the organelle fraction, and the fact that my experiment didn't work suggested to me that kinesin 1 was not the motor moving the vesicles in the organelle fraction or attached to and moving that particular species of vesicle. So now I had antibodies against the various kinesin proteins in squid, and I wanted to try to repeat the Western blot that Bruce did on the fractions of the sucrose density gradient. So once again, we have our gradient, and we have our cytosol, or the S1, which I renamed again one more time. I called it the supernatant. So I said supernatant, Bruce said cytosol, and sometimes it was referred to as the S1 in different publications. And of course, here we have our organelles that are also referred to as vesicles. And I first ran a gel. I mean, this is a Western blot, so again, we run a gel. So I ran supernatant, which is the S1, and organelles on a gel. And I just stained, as an example, stained the gel with Kumasi stain. So you see this intricate banding pattern of all of the different proteins in the supernatant and all of the different proteins in the organelle fraction. Then I did two different Western blots. So I ran an identical gel to this, and then I put it without staining it. I just ran the gel without staining it and laid it onto the nitrocellulose and transferred the proteins to the paper, to the nitrocellulose. And they're invisible, but they're on there. And I did soup and organelle again. Whoops. Organelle. And I probed this one with antibodies raised against the kinesin 3. And then I did a blot with soup and organelle that was against kinesin 1. So the same that Bruce ran. But this time I didn't dilute the supernatant. I just loaded an amount that gave me a nice band, and I loaded the same volume of the organelle fraction. So they're identical, and you can do a direct comparison in terms of amount. And kinesin 3, it turns out you really can't see anything. There's just like the faintest little band in the supernatant, and then in the organelle fraction, you get a really nice band for kinesin 3. And if you look at the kinesin 1, you get a really big band. I mean, kinesin 1 is smaller than kinesin 3, so it runs further down the gel because of its molecular weight. And I didn't detect anything in the organelle fraction, but that's fine. I mean, probably the detection mechanism that Bruce used, chemiluminescence, has a little bit more sensitivity than the assay I used. But at any rate, in this Western blot, it says that the kinesin 3 is in the organelle fraction and that the kinesin 1 is in the supernatant, or the S1, or the cytosol. So this gave me a clue that I might be onto something. Again, the kinesin 3 can't be in this fraction by itself. If it's just floating around, it will be in the supernatant because it doesn't have great enough density to get into the 15% sucrose layer. So it means that it has to be dragged down into this layer by something, and that something must be the organelles, must be the vesicles. So now I repeated my labeling experiment on 2 grids. These are the slides for the electron microscope. And of course I had vesicles. I mean, what you do is you put a drop of the vesicles on a piece of parafilm, and then the grid is really small. It's only 3 millimeters in diameter, and you can float the grid on the surface of the droplet, and then some of the vesicles stick, and you add the primary antibody, in this case kinesin 1, in this case kinesin 3, and then you have to add a secondary antibody. And if it's in an electron microscope, if you're going to look at the sample in the electron microscope, then you use a gold particle. And what we saw is no result, no interaction of the kinesin 1 antibody with the vesicles, but on the kinesin 3 sample, we detected kinesins, kinesin 3 on the vesicle. Both of these experiments are shown in figure 4 of the paper. Now we wanted to figure out whether the kinesin 3 antibody could inhibit movement of organelles along microtubules. So once again, we purified vesicles in our sucrose density gradient here, and we incubated the organelles with the antibody. So we have organelles and then we have the antibody. And the antibodies should, in theory, bind to the kinesin 3. So this is kinesin 3 antibody. And then we took a slide and we laid down microtubules. So you have a microtubule filament like this. And if you don't use the antibody, of course, the vesicles come down, they land on the microtubule, and then they move. So this is no antibody. But if you repeat the experiment, like this, and you have the antibody, what we saw is that the vesicle would come in and land, but it wouldn't move. So somehow the kinesin 3 antibody inhibited the movement of the vesicle. A further indication that kinesin 3 is likely to be the motor moving these vesicles along the axon. Finally, we wanted to go back into the axon and see whether we could study kinesin 3 in the cytoplasm. So we took our squid giant axon like this, of course with the terminal end of the cell, and our cell body and our nucleus with the DNA and our dendrites. And we dissected out the axon part of it, the giant axon, and we squeegeed out the cytoplasm by pressing a glass rod against the axoplasm and then pushing it along in this direction until we squeegeed out some of the axoplasm, the cytoplasm of the axon. And then we took our EM grid, which again is the slide for the electron microscope, which is a disc. It's a disc like this, and it has a grating on the disc like this, metal. This is metal grating, kind of like mosquito mesh for your windows. It's kind of like that. And then it's coated with a thin layer of clear plastic. And you lay the grid onto the cytoplasm, and then pull it off, and you end up getting a thin layer of cytoplasm on your grid. So if this is the grid, you just have a little thin layer of axoplasm. And that contains, of course, all of the different components, the microtubules, the organelles or vesicles, if you will, and then all of the other components that are found inside the axon. And then you can incubate the grid on a droplet of primary antibody, raised in this case against kinesin 3, and then a secondary antibody that has a piece of gold on it, and this is not drawn to scale. This is way out of scale. And we looked at it in a microscope, and we have a low mag image. This is shown in figure 6, and you can see this long, beautiful microtubule and a vesicle attached, and then it has gold particles in between the organelle and the microtubule. And then we have some close-up images of microtubules like this with vesicles on them. And indeed, we get labeling for kinesin 3 at the organelle microtubule interface. I mean, some of them are lower on the microtubule, like the gold particles down here. But, you know, the kinesin might extend down onto the edges of the microtubule, and the antibodies have some length to them also. So if the gold is just in this general region, it's likely that the gold is attached to a kinesin that's in between the organelle and the microtubule. We also know from other studies that kinesin 3s are faster, and they move at higher rates, similar to the 1.6 microns per second that's visualized inside the axon and the intact axon if you look directly like this, or if you look at extruded axoplasm laid on a microscope slide. So what do we take from all of this? Well, from my perspective, I think that conventional kinesin is not responsible for the movement of Ki-washed organelles along the surface of microtubules. I think that that's done by kinesin 3. So you biochemically purify the vesicles and you add them to microtubules. They move at 1.6 microns per second, and it appears to me that there's no conventional kinesin on their surfaces by electron microscopy. And if you biochemically purify the vesicles, most of the conventional kinesin is in the supernatant, 99% or something like that. Conversely, kinesin 3 is found on the surface of Ki-washed organelles. It's also found at the organelle microtubule interface in extruded cytoplasm placed on an EM grid. So, okay, it may be that kinesin 3 is responsible for those movements, but that leads to a whole variety of very interesting questions. And there's another paper that is in our folder written by Virgil Mirasan, who's wondering if kinesin 3 is responsible for the movement of those Ki-washed vesicles. What does conventional kinesin do? Because it's an extremely famous protein, and it's a really cool protein. And the discoveries that Ron and his team made in those days was just fantastic. But it leads the question to what is the role of conventional kinesin? And for that matter, what are the roles of the other kinesins that I discovered in my RNA database, in my cDNA library? Of course, conventional kinesin is a motor. There's no question about that. It's the founding member of the kinesin motor family. And we know that it moves because you can move glass beads along microtubules, or latex beads, of course, is what I meant, or you can get the microtubules to glide along the cover slips. So it certainly can generate force. It might still be an organelle motor, and that when you isolate the vesicles in the sucrose density gradient, and we add potassium iodide, and that could strip off conventional kinesin if conventional kinesin is more loosely associated with some subset of the vesicles, or the same set of vesicles as those that are tightly bound to kinesin 3. It could be that the different kinesin proteins move different types of cars, some moving Golgi vesicles, some moving mitochondria, some moving synaptic vesicles, and so on. There's still a lot of questions to answer, and it's a pretty fun topic of conversation. I'll end there. See you guys next time.